Welcome to our channel where biology meets clarity. Are you a CSIR NET aspirant looking to ace your exam? Or are you curious to gather deep knowledge and understanding in methods in biology? In this video, we will be diving into a detailed explanation of selected PYQs from Part C on methods in biology, specifically designed for CSIR NET exam. So, let's get started. Here we are with our first question, where we have to deal with some well-known biotechnique problems. And here we go. This is the first one. So it is stating about the radioimmunoassay. So in the radioimmunoassay, as a substrate, we generally use the radio-labeled isotopes. These isotopes are highly sensitive. Even it can be detected if it is present in a picomolar concentration. They are too much specific, hence the background noise is too low and they are highly intensive. So as a substrate, the radioisotopes are too much efficient to detect the target. So this should be the most efficient thing. Also, it has some health hazards. That's why today there is not that much use of this kind of things. And then next, it comes the chemi-illuminogenic substrates, CI. So this, what is this kind of substrates? So upon chemical reactions, this kind of substrates produces light. So like one of the example is the luminol, which is widely used in the case of ELISA, and which emits light when it gets oxidized by the HRP. And the next, it comes the chromogenic substance. The chromogenic substance. So this kind of substrate, one of the chromogenic substrate is the TMB, which is also is in use in the case of ELISA. So this TMB turns blue when it gets oxidized by the HRP. So see, in the case of chemi-illuminogenic substance, you can see it is emitting light, but in that case, it is just changing in color. It is producing some color. That means it is very much clear that this kind of things are too much efficient to detect the target. So in the first case, it is saying the RIA are more sensitive than the enzyme-linked assay where the chromogenic substances are in use. That is true. And next, it is saying that ELISA with chromogenic substrates are more sensitive than ELISA with chemi-illuminogenic substance. That is not true. And the next, it comes about the ELISpot. And what is the ELISpot? The ELISpot in the well plate, the capture antibodies will be there and will send a cell. So a single cell also we can send there. So this cell will secrete some proteins which will come here and bind here. In this test we have to determine the secretion of this cell. This is the target of this ELISpot test. So it is secreting some antigen, and that is coming and attaching with the antibodies, and there will be the second antibody, that will be a secondary antibody, will having a enzyme which will convert the substrate into a product. That is also happening in the case of ELISA. And this ELISpot, this measures the number of cells capable of secreting particular biomolecules. That is true. It is able to detect whether this is producing the single cell. Even each single cell from a population of cell is able to secrete the particular biomolecules which can be detected via this capture antibody. And thereafter, the secondary antibody will come and produces a product from the substrate. But this product should not be soluble in this solution that is present in the oil, because if it gets solubilized in the oil, then there will be no precipitation. There will be no spot formation. You will not be able to detect. So the soluble product, it should not be soluble product. It should be insoluble. So this is wrong. And the next one, in the case of Western blot, so like if this is the Western blot plate and your target protein is there, and again there you will send a antibody, there again there will be an enzyme, and here it will convert the substrate into the product. And here it is saying in the Western blot analysis, the product of enzyme substrate reaction. So in the enzyme and substrate reaction was here, where the protein is present, our target protein is present in the Western blot plate. There the antibody and enzyme, that complex should be there, and this enzyme and substrate complex, this enzyme will convert the substrate into the product. So the product of enzyme substrate reaction means that thing, that product reaction localized at the site of precisely where the antibody and antigen conjugate. Yeah, where the antibody and antigen gets conjugated means this part where this conjugation has happened, the signal should come from that side, because the product is the substrate. The product produces from the substrate. So so this should be the place where we should get the signal. From the product we will get the signal. If that is not happening in the antibody and antigen interaction place, then it will lead to produce the false positive result, and the noise should be very much. So it is saying that the enzyme substrate reaction should be localized, that product of the enzyme substrate reaction should be localized at the site of antibody and antibody and enzyme. That is the desirable thing. So this is right. So the option 2 is correct. So here a polypeptide sequence was taken and that was treated with some treatment procedures, and the results are given. You have to determine the primary structure of the peptide. So here, depending on the first treatment, the acid hydrolysis, here generally 6 molar HCl is used, and it can break any kind of peptide bond, whether the amino acid is present inside or at the N terminal or C terminal. Like if this is the chain, this is the polypeptide chain, and it is going through the acid hydrolysis, then all the kind of peptide bonds will be broken away. So only the amino acids will be available in the solution. So depending on this treatment, you can tell which kind of amino acids are present here. So let's go to the second option. So the aminopeptide is M. This is an enzyme, and depending on the name, you also can say the amino means it only can detect the free N-terminal end of a polypeptide. And peptidase, it also has a peptidase activity. That means it can break the peptide bonds. So if there is any kind of a free N-terminal end in the polypeptide, then only it can come and bind there, and it can show its peptidase activity. But here it is stating that it has not produces any kind of fragments. That means in our primary structure, there is no N-terminal is present there. But in all that kind of pictures, you can see the free N and C, both terminals are present. But in the last case, whether in that case the peptide is in a cyclic form, that means the terminal ends, if you say the lysine and arginine are on the terminal side, still there no any kind of N-terminal, C-terminal is free. They have engaged both the hands, both the sides to give a cyclic structure. So the option might be the fourth one. So let's see the third one. So here it is stating about the carboxypeptidase A and carboxypeptidase B. Here again the carboxypeptidase needs a free C-terminal end of an polypeptide, and then only it can show a peptidase activity. So and this kind of this CPA and CPB has a specific kind of residue range. It only cuts, that CPA only can cuts on the aromatic amino acids, or or if the branch aliphatic amino acids are there, like the tryptophan, tyrosine, or leucine, isoleucine. If those are present on the C-terminal domain, if in the polypeptide those amino acids are present and they are present on the C-terminal, then only they can come and cut. And also the carboxypeptidase B only can cut if there is a lysine or arginine on the C-terminal end. So here it has not produces any fragment. It is stating that sequence also might be the fourth one. Let's see the last one. So it is saying about the trypsin. So trypsin, it can detect only the lysine and arginine, and it can cut on the C-terminal domain. So let's say if this is the C-terminal domain, so the trypsin will come and cut this side, and here this will cut at this side. So thereafter there will be the two peptide chains like this and like this. The cut will be here. For that case, the cut will be here. So depending on this thing, and they have gone through the Edman degradation. They have gone through the Edman degradation. So, and there they got this and this sequence. So depending on all of the treatment's result, we can interpret that the answer is the fourth one. Let's come to this question. This is about a 3 kb circular plasmid. Let's see, this one is the 3 kb plasmid, the circular plasmid, and it was treated with different combination of restriction endonuclease. After treatment, they got this different kind of bands. Now say, if the BAM-H1 site is there, like they got two fragments. So in the circular form DNA, there must be two sites for the BAM-H1 recognition. So let's say this one is the one site, and it will produce a 0.5 kb length of DNA. So, and the rest will be the 2.5 kb band. And the next, so two restriction sites are there. The next one is the hinge 2, where you can see the 3 kb, the total 3 kb was there. That means there might be a single site which has transformed that circular plasmid into the linear one, or there might be no restriction site is there. That is also possible. That time the plasmid will be in that circular form. And the same happens for this also. There might be a single site, or there might be no site. Now go for the combinational part. So the EQR1 and BAM-H1 was used in a combination, and where they got the 0.5 kb band, this band is for the BAM-H1 site, which is also present when we have treated it with the BAM-H1. So now the EQR1 produces that 1 kb and 1.5 kb streak. So let's see, this is the 1 kb one, and the rest one is the 1.5 kb. So till that, this is 1.5 kb, and here this is the 1 kb from here to here. And let's see for the BAM-H1 and hinge 2. So here it has produces the 3 fragments. So one is 0.2 kb, another one is 0.3 kb, and the last one is the 2.5 kb. So 2.5 kb was also present when we have treated it with the BAM-H1. That means this is the BAM-H1 site part, and the rest one is the 2.5. So that 2.5 is present here, and the 0.3 kb and 0.2 kb means inside the 0.5 kb, that 0.5 kb band is not there. That means there must be a restriction site of hinge 2 inside that BAM-H1 site. So it must have produces a 0.3 and 0.2 site. So, and now let's go for the EQR1 and hinge 2 case. When it was treated with both the endonucleates, then it has produces a 1.3 kb stretch. This stretch is from this 1.3 kb, means this is the 0.3 kb, and this one is this green one is the 1 kb. So the whole total is the 1.3 kb. And here this is the 0.2 kb, and the rest one is the 1.5 kb. So this is the 1.7 kb now. So that means the structure should be like this, where the BAM-H1 site, this is the BAM-H1 site. Here in between there will be a hinge 2 site, and another side will be the EQR1, that is the so that should be the structure. And here let's see the answer part. So here the hinge 2 and EQR1 have a single recognition site each in the plasmid. That is true. Here you have to determine the incorrect words, incorrect sentences. So hinge 2 and EQR1 both are having a single recognition site. That is true. The next one is the hinge 2 site is located between two BAM-H1 site. Yeah, between two BAM-H1 site. That's true. The distance between EQR1 and BAM-H1 means that is the EQR1 and BAM-H1. The lowest distance between them is 1 kb. And the hinge 2 site and the BAM-H1, the distance between these two sites are this much, means 0.2 kb. It is 0.2 kb. It should be like this. So the distance between EQR1 and BAM-H1 site is less than between the hinge 2 and BAM-H1 site. This is wrong. And the last statement is saying the hinge 2 is located closer to one BAM-H1 site than the other. Yeah, this is the 0.5 kb site, and this is 0.2, and this is 0.3. So one site is more closer to the another cut site of BAM-H1. That is true. So the third option is the incorrect one. This question is based on some bioinformatic resources. Here in the column X, you can see the name of the resources, and in the column Y, the functions are there, and you have to match them. So the first of all, that TR-EMBL, so translational EMBL. So what is the EMBL? EMBL is actually a nucleotide sequence database, and just translating this database means that all the coding sequence present in that EMBL database, just translating that, we got the TR-EMBL resource. So that is a open resource. So it will go to the second one. This is the database of protein sequence, the simple one. And the next one is the T-BLAST-N. So BLAST-N is the basic local alignment search tool. That means it aligns in a local form, not in a global form. So here, so you have a sequence and you want to check whether it is having a alignment with any kind of known sequence or not. So here the T means the translation. So translational protein to N is nucleotide. So you have a translational protein sequence and you want to align it on nucleotide in a local basis. So this will go for the third option, the comparison of amino acid sequence against nucleotide sequence database. And the next one is the scope. Scope is this is the structural classification of protein database. This thing, the structural classification of protein database. So let's main target of this approach is to determine the evolutionary relationship between proteins, and it was widely prepared in a manual mode. So it should go to the scope, this one, the fourth one. And now the last one is the joint map. This joint map is a program for genetic linkage analysis, and we all know there is a relation between the recombination frequency and the linkage and the genetic linkage. So if the recombination frequency is too high, that means the linkage is low. So the option should be the first one. See this question. An investigator has identified a nuclear localizer signal in LS. This sequence is too much important to import the proteins that is present in the cytosol inside the nucleus. And you also can see the sequence that is full of basic amino acids. So at the C terminus of the protein X, so he has identified, like, if that is the protein X, then if this is the N terminal, this is the C terminal, you will get the NLS sequence on the C terminus. So the investigator fused protein X with GFP to analyze the localization of protein X. Analyze the localization, he has fused the GFP protein at this site. So that site means the C site. So we already know that if that is the NLS sequence, it will go inside the nucleus, but he wants to have a confirmation on that, so he has fused on the C site, the C terminus. So the fusion protein was detected in the cytosol, but he got this protein is present inside the cytosol, not in the nucleus. When the nuclear localizer signal was fused with JP at the N terminus, but if he is doing the same thing, this is protein X, this is the NLS site, the C site, and this is the N site, and here he has attached the GFP. Now he got the result that the NLS GFP extensively localized in the nucleus. This GFP is present in the nucleus, and you have to make the observation what is happening here. So at first we have to know how the NLS signal works. So like if this one is the outer membrane of the nucleus, and here you can see the pores. Those pores are known as the nuclear pore complex, and this is the nucleus site, this is the nucleus, this is the cytosolic site. And here this kind of pores are important in both the cases to import or export the proteins or mRNA, this kind of things. Like the transcription happens inside the nucleus, and thereafter after the maturation of the mRNA, it goes via the nuclear pore complex to the cytosol, where this mRNA produces the proteins. So the translation doesn't happen inside the nucleus, but the nucleus also needs some proteins for its function. So this kind of proteins, some specific proteins that the nucleus needs, it should go inside via the nuclear pore complex. So the importation should be there. And here, which kind of sequence are having this NLS, this specific kind of sequence, those only can go inside. And this sequence is a, if you see its structure, it is full of the basic amino acids. So how it works? This basic amino acid in physiological pH are having a charge, like they are having the positive charge. But in the importing proteins, which are too much responsible to import it inside the nucleus, that importing protein has a specific kind of amino acid sequence, which is the acidic in nature, and with in that physiological pH, it produces negative charge. So here the interaction happens between the NLS and importin. NLS and importin protein. So this kind of interaction is known as the electrostatic interaction. So depending on the interaction, that importin pulls the that NLS and its sequence, like this kind of S proteins inside the nucleus. But now you have to tell what is happening after understanding the nature of this NLS and its mode of action, how it goes inside. So so in the option A, it is saying the basic amino acid in the protein X GFP. So basic amino acid means the NLS sequence is masked by the GFP sequence and thus not capable of directing the entry of the protein X GFP in the nucleus. It can happen because this GFP, after some modifications, it can go and binds it, like in the protein interaction. Like if you have to make the interaction between the NLS and protein, that NLS portion should stay at the outside of that protein structure, or else if it is going inside, it will not be detected by the protein. So must be the GFP is doing some work here. Must be it is masking the NLS sequence, which is for that it is not able to go at the surface area, and that important protein is not able to detect it. So it might be an option. That might be a right option. And the B option is saying about the X protein in the full-length XGFP chimeric protein is post-translationally modified that impacts its import in the nucleus. In that case, in the second case, and if this is the first case, the second case, we can see this protein is going inside. There is no problem. That means the post-translation any modification like phosphorylation, methylation, acetylation, these kind of things are not happening for this X protein part. Because this protein can successfully be introduced inside the nucleus in the second case, but not in the first case. So must be something is happening at the GFP part, because this is in the where it is present at the C terminus. So there is no translational any modification in the case of X protein. So it must be wrong, but it can happen in the case of GFP, because after the binding of the GFP, it is changing its structure. It is not allowing the NLS to go on the upper part. So that means there must be some modifications which is masking the NLS. The modification must be in the GFP part, because in the second case, it has introduced, it goes inside the nucleus. So this option D might be right, because it is stating about the modification at the GFP side, the post-translation modification at the GFP side. And the C option, that is saying the fusion with GFP makes the protein X too bulky. So in first and second, in both the cases, the GFP protein is there. So it can't like change the molecular weight that much. So bulky, that is interrupting it from entering inside the nucleus. That must not be the true thing. So the correct option is option 1.